Hello students, we will continue with chapter 5, that is the fundamental unit of life, and we have studied till here till the basic structure of the cell. What is a cell we have studied in the last topic, and now today we will start from the fresh topic, that is what is a cell made up of. See, if you observe a plant cell or an animal cell or any other cell, then you must have seen that there are three common features on all types of cells. The first common feature is the plasma membrane, the nucleus, and the cytoplasm. So these are the three features that are common to every cell. The cells have special components called cell organelles. Inside the cell I'll show you the cell. Inside the cell there are certain components which are called as the cell organelles, which perform different functions. And three cell organelles are common to all types of cells. All the activities inside the cell and interactions of the cell with the environment are possible due to these features. That means whatever the activities that are going on inside the cell, and when the cell is interacting with the outside environment, these three features are common to them. Plasma membrane is involved in it, nucleus and the cytoplasm is involved in these activities. Okay, now let's move further. Today we will be studying about the structure of plasma membrane in detail. The plasma membrane, it is also called as the cell membrane. Okay, so what exactly the cell membrane is? The outermost layer, it is the outer layer which forms a partition between the inner cell components and the outside environment. Okay, cell membrane or the plasma membrane, it encloses all the components of the cell, so therefore it separates the inner environment of the cell with the outer environment. It keeps the cells, all the components of the cell together, separated from the outside world. So what the cell membrane is, it is the outermost covering of the cell that separates the contents of the cell from the external environment. The plasma membrane allows or permits the entry and exit of only some materials in and out of the cell. This point is very important. That means the plasma membrane in a cell is not allowing everything to move inside and outside. It is allowing only some material, only particular material to move inside and to move outside. So therefore the membrane, it is also called as selectively permeable membrane. Plasma membrane, therefore it is also called as the selectively permeable or semi-permeable membrane. Okay, now we'll see further. This is the diagram of the plasma membrane. It is made up of lipid bilayer, and proteins are also present in the plasma membrane. So the movement of particles, movement of substances, either it is taking place across the proteins or through the lipid bilayer, the movement of substances is taking place. Okay, so here we can see that this is the fluid mosaic model of the plasma membrane. See, the plasma membrane is not a rigid structure, it is flexible, so that is why the term fluid mosaic model is used for this plasma membrane. So here in class 9th you must have an idea that the plasma membrane, it is made up of the lipid bilayer and it has proteins also across which, through which the movement of particles take place. Some substances like carbon dioxide or oxygen can move across the cell membrane by the process of diffusion. Now I'll tell you the process of diffusion. I'll explain it to you first, then we will read. Suppose I've taken a beaker and I have added a red ink to one of the corner of the beaker which contains water. Now after some time, what will happen? The ink, it would spread uniformly throughout the beaker in this manner. So what is exactly happening in this process? There is movement of particles from high concentration to low concentration. until the concentration becomes equal. Okay, so this process is called as diffusion. Now what the definition of diffusion is? The movement of particles, yeah, movement of substances from high concentration to low concentration. So the substances like carbon dioxide or oxygen can move across the cell membrane by the process of diffusion. I'll explain you again this thing with the help of the diagram. This is a, let's say, plant animal cell. Here oxygen is the concentration of oxygen is more towards the outer side, and concentration of oxygen is less in the on the inner side. So from higher concentration to lower concentration, the movement of oxygen would take place. Similarly, carbon dioxide is formed as the waste material inside the cell, so the concentration of carbon dioxide is more towards the inner side. So from high concentration, the carbon dioxide moves towards outside where the concentration of carbon dioxide is low. So this is how the movement of carbon dioxide and oxygen is taking place across the plasma membrane. Okay, now let's see the definition of diffusion once again. The spontaneous movement of a substance from a region of high concentration to a region where its concentration is low. Something similar to this happens in the cell. For example, substances like carbon dioxide accumulates in high concentration inside the cell. This point I have explained you. In the cell's external environment, the concentration of CO2 is low as compared to that inside the cell. So as soon as there is difference of concentration, that means if there is a large concentration of carbon dioxide inside the cell, then carbon dioxide from inside it would move to outside where the concentration of carbon dioxide is low. Then this process is called as diffusion. Okay, now we'll learn one more term here, osmosis. First, try to understand what exactly osmosis is. Suppose I've taken a beaker and I have placed a semi-permeable membrane in the middle of the beaker. I have fixed the membrane in the middle. Now, this is the semi-permeable membrane. That means it is going to allow the movement of only some particles across it. Not everything, only some of the particles across it. Okay, semi-permeable membrane. I'll give you one example. You must have seen a boiled egg. When you remove the shell of the boiled egg, then there is a very thin membrane. So that is the example of a semi-permeable membrane. So that can be used here in this particular experiment. Now, when I have placed the membrane here, then what do? I put two types of solution here. One is dilute solution, the other one is concentrated solution. Okay, I hope you understand the difference between the two, dilute and concentrated solution. Okay, and before moving forward, I'll explain you one more thing: solute, solvent and solution. Suppose I take a beaker, I add water to it. So water is the solvent. Water is the solvent. And if I add, let's say, 2 teaspoons of sugar to it, then sugar is the solute. And if I mix both of them, then what is formed? Solution is formed. So these three terms should be very clear to you: what is solvent, what is solute and what is solution. Okay, water is solvent. If we are adding sugar or salt to it, that is the solute, and together when it is getting mixed, it is getting dissolved here, then we call it as solution. So here it is less concentrated solution, here it is more concentrated solution. In order to reach equilibrium or to balance the solution on both the sides, either the solute should move from this side to this side, or the solvent should move from this side to this side. ठीक है, दोनों में से एक प्रोसेस होना चाहिए. In order to equalize the concentration, either the solute should move or the solvent should move from this side to this side. Now because there is a semi-permeable membrane, it would not allow the movement of solute. it would not allow the movement of solute. Therefore, the solvent, which is water, it moves from this side to this side. And after some time, this situation comes when water has moved from this side. That means, let's call it as A side to B side. Water has moved from A side to B side to equalize the concentration on both the sides, and we can see that the level of water has risen up on the B side, and the concentration is now equal on both the sides. So this process is called as diffusion. Now how do we define it? This process is called as osmosis. And how do we define it? Osmosis is the diffusion of water molecules. Okay? Osmosis is the diffusion of water. We can see that water is high in concentration here and water is in low concentration here. So water is moving from high concentration to low concentration. So we can say osmosis is diffusion of water. Okay, or we can define osmosis as the movement of water from its high concentration to the level of low concentration through a semi-permeable membrane is called as osmosis. Okay, so I'll again repeat both the definitions of osmosis. Osmosis is the diffusion of water through a semi-permeable membrane. The second definition is the movement of water molecules from their higher concentration to their lower concentration through the semi-permeable membrane is called as osmosis. Okay, so the movement of water molecules through selectively permeable membrane is called osmosis, and the second definition it is given here. The osmosis is the passage of water from a region of high water concentration through a selectively permeable membrane to a region of low water concentration कब तक till equilibrium is reached. Okay. What will happen if we put an animal cell or a plant cell into solution of sugar and or salt and water? See, before proceeding further, I'll explain you the difference between hypertonic, hypotonic and isotonic solutions. You listen to it very carefully. There is a beaker. Let's say there are two beakers, A and B. Okay, I have taken water, equal amount of water in both the beakers. So we add the water in both the beakers, equal amount of water we are adding to both the beakers. Now let's say we add 2 teaspoons of sugar in this particular beaker, beaker A, and let's say we are adding 7 teaspoons of sugar in beaker B. Now beaker B is going to have more concentration as compared to beaker A. So if we compare both the beakers, then we can say that beaker A is hypotonic. Okay, you should be very clear with these terms. Hypo means less. Okay, so beaker A is having less concentration in comparison to beaker B. So therefore we call this as hypotonic solution. Beaker A has hypotonic solution in comparison to beaker number B. Now the beaker number B will be called as having hypertonic solution because it is having more concentration in comparison to beaker number A. So we call this as hypertonic solution. I hope both the words are clear to you. The solution having more concentration is called as hypertonic and less concentration is called as hypotonic solution. Suppose there are two beakers which have equal concentrations. Then we say that both solutions are isotonic to each other. Iso means same. So we say that if both the beakers have similar concentration, similar amount of water, similar amount of solute in it, let's say 5 teaspoons of sugar here and 5 teaspoons of sugar here, then we say that both the solutions are isotonic in nature. Okay, now we will proceed further. What will happen if we put an animal cell or a plant cell into a solution of sugar or salt water? One of the following three things could happen. If the medium surrounding the cell has higher water concentration than the cell, meaning that the outside solution is very dilute, the cell will gain water by osmosis. Such a solution is known as hypotonic solution. Now you try to understand this particular situation. There is a beaker, and in this particular beaker we are keeping a cell. It could be a plant cell or an animal cell. Okay, we have placed a cell here. So if the medium, this solution that we are taking, if it is having less concentration, then it would this plant cell or the animal cell, it is going to absorb water from the outside solution. So this solution is called as hypotonic solution. Why it is absorbing water? Because the concentration inside the cell is more and concentration outside the cell is low. So from low concentration the water would move, here the concentration of water is more towards the outside. So from outside the water would move towards the inner side, because we are concentrated on water concentration here. Water concentration is more towards outside, so from outside it would move inside the cell. Okay, so if the medium surrounding the cell has high water concentration than the cell, the meaning that the outside solution is very dilute, the cell will gain water by osmosis. Such a solution is called as hypotonic solution. I'll again repeat once again. There is a cell in a beaker, let's say, and the solution is hypotonic. So here the concentration of water is more on the outer side. So from outside the movement of water would takes place inside the cell. So this happens in case of hypotonic solution. Now the second situation is water molecules of if the medium has exactly the same water concentration. See, we've taken a beaker again and we've placed the cell here. Now the concentration of the cell and the solution is same, so there is no net movement from outside to inside or from inside to outside. So such a situation appears in case of isotonic solution. So water molecules are free to pass across the cell membrane in both directions, but more water will come into the cell than will leave. This we are talking about hypotonic solution here. When the concentration of water is more towards outside, it would move inside the cell, and the net result is that water enters the cell and the cell is likely to swell up. Okay, when we place the cell in hypotonic solution, the water would move towards the inner side of the cell and the cell would swell up. But in case of isotonic solution, neither the cell would swell up nor it will burst. It will remain as it is, because the concentration of the cell and the concentration outside the cell are same. Here you can see with the help of this particular diagram, this is a cell, this is the animal cell. When concentration of water is more towards outside, its movement would take place towards inner side and the cell would swell up and it may burst also. Here it is isotonic solution. The water would move towards outside and it would move towards inside also, and there is no net change in the shape of the cell. Here in case of hypertonic solution, the water moves outside because in hypertonic solution the outside medium is having less concentration of water, more solute but less water. So water from the cell would move towards the outside and the cell would shrivel, or we can say the cell would shrink. So this happens in case of animal cell. Okay, so we'll see the third situation also. If the medium has low concentration of water than the cell, meaning that it is very concentrated solution, the cell will lose water by osmosis, and such a solution is called as hypertonic solution. I will explain it to you once again. There is a beaker, let's say we have placed a cell in the beaker and the beaker is filled with very high concentrated solution. Now concentrated solution has less amount of water and the cell has more amount of water, so water would move from the cell to the outside solution, and as a result this cell would shrink. And this shrinkage of cell is happening in which case? In case of hypertonic solution. So again water crosses the cell membrane in both directions, but this time more water leaves the cell than enters it, therefore the cell will shrink. So thus osmosis is a special case of diffusion through a selectively permeable membrane. Now let us try another activity. See, I've explained you all the three situations. If the cell is placed in hypotonic solution, then it would swell up. If it is kept in isotonic solution, it will remain as it is, and if it is kept in hypertonic solution, then it would shrink. Now let's move further. There is one activity that is given here. In this particular activity, what is done? We take remove the shell of the egg by dissolving it in dilute HCl. We take an egg, we keep it in dilute hydrochloric acid, and what do we see? The shell, as it has calcium carbonate, it would dissolve, and only outer skin, that is the membrane, only it remains the egg. It encloses the egg. Calcium carbonate is removed. Now what do? We put this kind of an egg in pure water and observe it. Okay, if we observe it, if we keep it in water, then what would happen? After some time the egg would absorb water and it would swell up. Why it is happening so? Because towards the outer side the water concentration is more, and inside the egg water concentration is less, so from outside the water would move towards the inner side. This is the de-shelled egg. Okay, now place a similar de-shelled egg in concentrated salt solution and observe for 5 minutes. The egg shrinks. Why? See, if we keep the de-shelled egg in concentrated solution, either it is concentrated sugar solution or concentrated salt solution, then what do we observe? The water from the egg will move out towards the outside, because water concentration is more towards inner side as compared to outside, as we have taken it in the hypertonic solution, which was having more concentration. Okay? So as a result the egg would shrink. Water passes out of the egg solution into the salt solution because the salt solution is more concentrated. Okay, we can try this activity with dry raisins or apricots also. In the next activity they have shown you that if you keep the dry raisins or dry apricots in plain water, they will absorb water and they would swell up. But if you keep the raisins in concentrated sugar solution or concentrated salt solution, then they will shrink. Okay, next, unicellular freshwater organisms and most plants tend to gain water through osmosis. That means that when the water movement is taking place in unicellular organism, it is taking place through the process of osmosis. And absorption of water, when it takes place through the plant roots, it is also the example of osmosis. So diffusion is very important for exchange of gases, for exchange of water in the life of a cell. In addition to this, the cell also obtains nutrition from the environment through the process of diffusion only. Different molecules move in and out of the cell through a type of transport requiring use of energy. This I'll explain you later. See, there are different molecules which have to be transported uh, against the concentration gradients. Therefore, they require energy. Okay. The plasma membrane is flexible and it is made up of organic molecules like the lipids and proteins. That I've told you, I've shown you the diagram of the plasma membrane. Proteins and lipids are present in the plasma membrane and it is not a rigid structure, it is highly flexible. However, we can observe the structure of the plasma membrane only through the electron microscope. So, after the discovery of electron microscope only, it was possible to observe the different cell organelles. The flexibility of the cell membrane also enables the cell to engulf in food and other material from its external environment. Such processes are called as endocytosis. See, in case of certain organisms like amoeba, the outside food, it is taken in by through the surface of the cell, and it is possible the pseudopodia are formed, and it is possible because the membrane is flexible. If it was not flexible, then this process would not have taken place, and this process of acquiring food in this manner through the membrane, it is called as endocytosis. So amoeba acquires its food through such processes. Now they've given one more activity. Find out about electron microscope from the resources and discuss it with your teacher. Okay, this activity you can do it on your own. Okay, now let's see the questions. How do substances like carbon dioxide and water move in and out of the cell? They move through the process of diffusion. You will write the answer that oxygen is more on the outside, so therefore from the outside it would move towards the inner side by the process of diffusion. And carbon dioxide is formed as the waste material. It is high in concentration inside the cell, so from inside it would move towards the outer side. Okay, why is plasma membrane called selectively permeable? Because the answer is very simple, because it does not allow everything to pass across it. It allows only selected particles to move across it. Therefore, it is called as selectively permeable membrane. So with this we come to the end of this particular topic, cell membrane or the plasma membrane. Tomorrow, next turn onwards, we will be studying about the cell wall. So that's it. Thank you.